Welcome, future physicians. I'm your lead geneticist, and in today's explainer, we're getting right down to the molecular bedrock. We're going to uncover how the microscopic anatomy of DNA literally forms the absolute foundation of the clinical diagnostics you'll be ordering every single day. Let's get right into it. Okay, let's take a quick look at our roadmap. We're going to hit 5 key areas today: the clinical genetics landscape, early theories of inheritance, transformation, cracking the genetic code, and finally, the anatomy of the double helix. Starting off with part 1, the clinical genetics landscape. So your future diagnostic toolkit is basically split into these distinct branches. You've got cytogenetics and molecular genetics for spotting structural stuff for specific inherited syndromes, but look at pharmacogenetics and oncogenetics. This is the cutting edge, right? If your patient has melanoma, understanding their specific tumor mutations at the molecular level is what actually lets you prescribe a targeted BRAF inhibitor instead of just, standard scattershot chemotherapy. It all starts right here. Moving on to part 2, early theories of inheritance. To really appreciate the tools we have now, you kind of have to look at the crazy historical detective work it took to get here. I mean, we've made a massive leap. Think about the ancient, highly flawed theories. Pythagoras was pushing spermism way back in 530 BC. Aristotle thought it was this mutual mixing. And get this, by 1520, Paracelsus was actually pushing preformism, literally the idea that there was a homunculus, a tiny, fully formed little person just chilling inside a sperm cell. Contrast those wild guesses with our modern reality: Mendel's inheritance laws, Sutton and Bovary's chromosome theory, and Watson and Crick mapping DNA in 53. We went from tiny imaginary people to precise physical mapping. All right, part 3, transformation or foundational case. This timeline is absolutely crucial because it traces our shift away from a massive scientific blind spot. For the longest time, top scientists swore that complex proteins were the hereditary material. But starting with Mischer isolating nucleic acids in 1869, we see this slow, 80-year investigative slog. Through Griffith's work in 28, Avery and his team in 44, and finally Hershey and Chase in 52, we finally zeroed in exclusively on DNA. Let's walk through Fred Griffith's brilliant 1928 protocol. This is the experiment that changed everything. He worked with a virulent S strain of Streptococcus pneumonia that killed mice, and a harmless R strain. He injects heat-killed S strain. Mouse lives, obviously. But step 4? Well, that's where the magic happens. He mixes the dead S strain with the live R strain and boom, the mouse dies. He proved that genetic traits can literally be physically transferred. Miraculously, that harmless R strain sucked up some invisible chemical substance from the dead S strain and became completely lethal. We call this transformation. And for you as clinicians, this isn't just old history. This exact experimental groundwork is the engine for all modern biotechnology. It's literally how we engineer bacteria to pump out human insulin and antibiotics today. A real game changer. Then we jump to 1952. Avery's team had already pointed the finger at DNA, but the skeptics were still holding out for proteins. So Hershey and Chase shut down the debate for good. They cleverly used radioactive phosphorus-32 in bacteriophages, viruses that infect bacteria. Since DNA has phosphorus and protein doesn't, they could track exactly what the virus injected to hijack the cell. Bam. Conclusively proved it was DNA, not protein. The debate was officially over. Which leads us to part 4, cracking the genetic code. Once we knew DNA was the culprit, the next massive hurdle was figuring out its chemical anatomy. Enter Erwin Chargaff in the early 50s. He figured out that the amount of purines always perfectly matches pyrimidines. A always pairs with T, and C always pairs with G. In human cells, adenine and thymine are about 30.3% each, while guanine and cytosines sit around 19.7%. And that ratio, strictly species-specific. Now, why should you care? Because Chargaff's rules aren't just trivia. This predictable, complementary base pairing is the exact physical property that makes your PCR and diagnostic sequencing tests work. It's why those lab protocols are so incredibly accurate. And we absolutely have to talk about Rosalind Franklin's famous X-ray diffraction photograph. You see that distinct cross-like pattern right in the center? That provided the vital, missing structural data. Without this exact image, the rest of the team wouldn't have been able to mathematically deduce the 3D shape of the double helix. She completely hit the nail on the head. But before we look at the final Nobel Prize-winning model, we really have to ask ourselves a fundamental question. How does this incredibly simple chemical alphabet, literally just 4 letters, dictate such complex biological function?
How does this basic molecule control every single human trait and disease you're going to treat?
Let's find out in Part 5, Anatomy of the Double Helix. Okay, let's dissect the core building blocks, the nitrogenous bases. Notice the structural difference here. We've got the big double-ring purines, adenine and guanine, and the smaller single-ring pyrimidines, thymine, cytosine, and uracil in RNA. Think of it like a ladder. For the DNA ladder to stay the exact same width all the way down, a large double-ring purine always has to pair with a smaller single-ring pyrimidine across the center. That is structurally why A pairs with T and C pairs with G.
Looking at the full architecture, the basic unit is the nucleotide, a phosphate, a pentose sugar, and a base. The two strands are anti-parallel, so one runs 5' to 3', the other is essentially upside down, running 3' to 5'. They are locked together by a super strong sugar-phosphate backbone linked by phosphodiester bonds. But, and here's the genius part, the bases in the middle are held by weak hydrogen bonds, 2 for AT, 3 for GC. It's a brilliant evolutionary design. The primary sequence is securely held by the backbone, but those weak center bonds mean you can unzip the helix like a jacket to copy it or melt it apart in the lab for your diagnostic assay.
Basically, think of these 4 bases as a simple chemical alphabet. Just like letters make words, the specific order or sequence of these nucleotides dictates the primary structure. This sequence holds the absolute blueprint for all cellular proteins. A single, tiny typo in this primary sequence, that's exactly what causes the inherited disorders you'll be diagnosing, like sickle cell anemia or cystic fibrosis.
And just to put the sheer scale of your clinical task into perspective, look at this number. The human genome contains 3 billion base pairs of this chemical alphabet. 3 billion letters that have to be perfectly copied every time a cell divides, which means 3 billion places where a tiny molecular error can trigger a major clinical syndrome.
So from ancient Greek guesswork to early bacterial experiments, the whole shebang culminates right here, in the routine genetic lab tests you're going to be ordering. Whether you're using cytogenetics to look at whole chromosomes or molecular tests to hunt down a single point mutation, you are directly relying on those base pairing rules and that double helix structure discovered decades ago to confirm a specific inherited disorder.
So, as you step out of the classroom and into your clinical rotations, I want to leave you with this provocative thought. Knowing exactly how the DNA molecule was discovered and precisely how its structural alphabet works, how will you leverage this ultimate clinical detective tool to change a patient's life tomorrow? Keep that in the back of your mind as you study. Thanks for joining me on this explainer, and I'll catch you in the next one.